We welcome you back for today's second session. The moderator for this academic session is Dr. Sunil Kumar, Associate Professor AINN. The reporter is Ms. Gursimran, MSc Second Year AINN. Taking the session further, we would like to introduce Dr. Chetna Sachidanandan, Head, Molecular Biology Division, a distinguished researcher at CSIR Institute of Genomics and Integrative Biology. Her work encompasses a broad spectrum of molecular and structural biology with a particular emphasis on deciphering the molecular intricacies underlying various developmental stages and diseases states. Her contribution have garnered widespread acclaim with the scientific community, earning her several prestigious awards and accolades. Among her notable honors are CSIR Young Scientist Award, recognizing her outstanding contribution to scientific research, and the Deputy Overseas Association, which highlights her international recognition. I'm extremely sorry for that. I would like to continue. Okay. Okay, her impact extends beyond the laboratory as evidenced by her prolific publication record, which highlights over 50 peer-reviewed articles in top-tier scientific journals. These publications not only showcase depth of her expertise, so the topic of the session is zebrafish as a model for human neurodevelopmental and neuropsychiatric diseases. Ma'am, we would like to request you on the stage.
Use this or
you can use this. You can use
this doesn't work?
No, that doesn't.
Okay. Okay, can you hear me without this? Do we need? Okay. Okay. All right. Yeah. Works. So this is the age of the hybrid talk and online meetings, right? So, all right. So this is what I'm going to talk about. And at the outset itself, I must say your department is a neuroscience department, and this workshop is a very intensive neuroscience workshop. I feel like a little bit of an imposter here because I'm not a neuroscientist. If you'll see my work, you will realize that I am aspiring neurodevelopmental biologist. So we use zebrafish as a genetic model to understand diseases that affect the brain and other organs. And so I thought what I would do during this talk is also to introduce you to the sort of the genetic potential, the great things that you can do with this model. I'm sure you have done already practical experiments, behavioral experiments with zebrafish. I think that's you've done some in the course, right? Right. So instead, I will talk a little bit about what all kind of molecular inquiries you can do using this system and why it is such a powerful system. And I'll start with a little bit of introduction about the fish as a model and go on to tell you a story of one of our research work. So this fish comes from India. It is now used as a developmental model all over the world, but all these are the sort of the places where you see the fish commonly. And many Indians who live in rural areas are very familiar with this fish. This is not new for them. Will this work? Okay. All right. So no, this is fine. I'm standing here only. So इसके अंदर होगा. So I'll continue talking. So the fish is a model for developmental biology because of certain advantages, and it will become clear to you as I talk. But sort of illustrate the beauty of the young tiny embryo that you can actually start observing from one cell stage. And this is something unique when you compare to mammalian systems, because in mammals, to get access to the early stages of the embryogenesis, it is not that easy, right? The fertilization happens internally, and you will have to sacrifice the mother or surgically remove the fetus or the embryos from inside. And that is a very difficult procedure, as well as there is a lot of guesswork involved in what the embryonic stage would be. So here, zebrafish males and females mate externally. So basically, the female lays the eggs and the male will fertilize it externally. So you can pick up the embryos the moment fertilization has happened, and you can start observing them from the zygote stage. That allows you a beautiful window into development that is not possible in any other vertebrate, right? So, yeah. So, and if you want to use it as a model, is it comparable to humans? So let's look at all the organs that are placed here. So this is a larval stage, and this is an adult stage, right? So adult is only around 3 to 4 centimeter long. And the larval stage itself, by 2 days, all the organs in the animal have sort of been placed in the right places, and the developmental processes have gone on. Your patterning has begun. After around 2 days or so, all you see is the elaboration of these organs. So they grow in size, they become more complex with time. But in the first 24 to 48 hours, all the systems that will exist in the adult are already determined. Cells are in the right place. Differentiation have happened, right? So great system for looking at development and for looking at the brain too. So this is just a comparison to see the sort of the placement of different structures in the brain in zebrafish. And we have a full complement of all the parts, all the subsections in the brain. Of course, there is a huge difference between the proportion of cortex in human to fish. So just a little bit of historic sort of perspective on how fish became a model. So this is around the 80s or so, when George Streisinger picked up from the pet shops in Oregon. So if you walk around the pet shops within India, in Delhi itself, you will find that most pet shops will have zebrafish in them. And that's because these fish are really robust. You don't have to do a lot of sort of caring for them. And they are quite a hardy fish, easy to maintain, and they even breed in captivity, which is also very difficult for many organisms, right? So he picked up these fish, started kind of fiddling with it, seeing whether he can set up breeding and collect embryos in right time, etc. And Charles Kimmel kind of picked it up and made it more of a developmental model. But the real explosion of this field happened when these three people, so Christian Nusslein Vollard, Mark Fishman, and Wolfgang Drewer, created a set of resources for this organisms research. And what they did was, so Christine Nasrin Wollard is a Nobel laureate who got a Nobel Prize for doing a huge genetic screen on Drosophila and identifying the first sort of set of genes that we understand with roles in development. Those, the developmental genetics of Drosophila really kind of led the way in understanding embryonic development. And once she had done that for Drosophila, she decided great strategy to do for fish too. So this is what they did. So how do you do forward genetics? What is forward genetics? For those not familiar with it, basically we create mutations randomly in an organism with no clue of where we are mutating, and we pick up interesting phenotypes. So if I am interested in eye development, I would pick up the animals where the eye development is going wrong, right? So you're picking up places where something that was important for eye development did not work properly. That's your definition. Pick up those fishes. So you would mutagenize the male. The sperms of the male would get mutated, the gamete. You would cross it with a wild type female and then outcross a number of generations to get clean mutant lines. So when you look at these mutants, you have a interesting phenotype to guide you, but you have no clue where this mutation is. In those times, when they started doing this genetic screen, we did not have a map of the genome also, right? So you would do something called chromosome walking and linkage mapping to figure out which genes are mutated. This is a very painstaking, laborious process. So each postdoc in the lab would pick up one mutant and sort of take it back, walk back to the genome to figure out of this huge number of chromosomes and the whole genome, where is this mutation that is causing this phenotype that you're seeing, right? So this was how genetics was done. And this is called forward genetics. But kind of after this whole exercise was done, a huge number of mutations were generated, and a large number of labs were born out of this effort. Okay. So as I said, each postdoc picked up one interesting developmental area that was fascinating to them. Suppose it is brain development to you. So things that are the fishes that have problems in brain development, that postdoc picked up and went and started a lab of their own. So like this, there were number of labs born out of this huge screening effort that happened. But as the genome sequencing processes, kind of the technology came, the money was available, and people started looking at the genome. So did the zebrafish biologist. The zebrafish genome came out much later than the human genome, not because it was not done, but there were a huge number of repeats and it was never completed. It is still not fully complete. So there was a lot of delay in publication, but finally the community decided we just need to put it out. Although everybody was using the sequence that had been put on the database already, right? So the paper came out and we discovered that there are 70 to 80% genes in humans have ortholog in zebrafish. More than one ortholog, actually. So it was also discovered that sometime during the evolution, teleos had a genome duplication. And when you look at the zebrafish genome for genes that are of interest to you in humans, say, the orthologous gene there, you would find 2 copies of many of these genes, and usually on 2 different chromosomes. And that is because the whole segments of chromosomes got duplicated and split into different regions of the genome. So, we can find orthologs of 70 to 80% of human genes in fish. And this was the laboratory strain of fish that was sequenced. This is the Indian strain of fish that was caught from the wild fish that was caught from Assam. This sequencing was done in our institute, CSIR IGIB, by a group of scientists headed by Dr. Sridhar Shivasubbu and Vinod Karia. So they published the, around the same time, they published the Indian version or the wild version of the zebrafish genome. Comparisons show that the wild version have some interesting differences, which is not unexpected because laboratory strains are protected in a protected environment for generations, right? So the wild animals have to kind of deal with infections, predators, and the wild atmosphere or the environment for a longer time. So there are quite a few differences in the genome that you see. Anyway, with these two genomes now available, what would you do? Would you go and do a forward genetic screen, create random mutations? It no longer, right? I don't think any of the people sitting here do that kind of genetics anymore. This is what we do now, what we call reverse genetics. So now we know the genes, the sequence of the genes, where the genes are placed in the chromosome, how many copies are there changes in them? We know all this background information already, right? So with this information, now what scientists do is they pick up genes that are of interest to them, usually guided from what is known in human or Drosophila or other organisms. So a gene that has popped up in a disease study, for example, right? Something that looks very important to you, you want to mutate that gene. And how would you mutate or basically perturb that gene? So you want to understand what the function of that gene is and how it correlates to phenotypes that you see in the organism. So one way, the sort of the quickest and easiest way to look at gene function in zebrafish is to transiently knock down a gene. And by this, what I mean is that for a short period of time, you can reduce the levels of the gene product in the embryo. And this is easiest done when you can access the one cell stage embryo, because I can inject DNA, RNA, chemicals, proteins, whatever I want into the one cell stage. And when I inject it into the one cell stage, every cell of that embryo will get that product, right? So what I'm going to show you here is how you use an antisense oligonucleotide to knock down genes or reduce the levels of genes, right? So let me, so this is how an injection would happen. Have you guys seen injection in this course? Did you do any micro injections? Okay. So what you would do is you can design oligonucleotides that are antisense to the mRNA of the protein that you're interested in, right? So let us look at two strategies in which you can do that. The most sort of simple way of thinking of it is, I make an antisense oligo that binds to the region where the ribosome initiates translation, right? The start codon and its sort of neighborhood. So if I create an oligonucleotide against that region, what will happen is when I inject it into the one cell stage embryo, the oligo would go and bind to the mRNA initiation codon and its area. Ribosome is not able to recognize that sequence and go and bind there, right? So when the ribosome is not binding, the protein will not get made. And this will not happen in 100% of the mRNA, okay? So even if I inject large amounts of oligonucleotide, there will be some escapers. So basically what you would have is a reduction in the protein levels because the translation is impaired. That's one way of doing it. Another way of doing it is that I would design an antisense oligo to a splice junction. So you know that splicing happens when huge protein complexes called spliceosomes go and bind to the splice junctions, acceptor and the donor sites, recognize them, bring them together, cut these two pieces and stitch them together, right? This is a process that happens when splicing happens. So if I make an oligo against one of those sites, again, the spliceosome machinery, the proteins will not be able to bind there and your splicing will not happen. When the splicing doesn't happen, what you have is, instead of splicing 3, 4, and 5 exons, you would have the intron trapped inside. It will not get spliced out. When you have an intron here, the translation machinery, when the ribosome keeps reading, it will reach somewhere here. And what happens in introns is that you will find lots of stop codons there. So the ribosome will stop there and you will have a smaller protein compared to the wild type, right? So this is one way to reduce the levels of protein. But what if I want to mutate or remove the protein coding gene altogether? So this is again new technology that was not available even when I was doing postdoc like 10, say 12 years ago or so. The CRISPR technology really exploded in the scene of biology in the last maybe 5 to 7 years, right? when it became so easy to use that now anybody can do CRISPR mutations, right? So we have three different technologies. So CRISPR was not the first genome editing tool that came into being. Zinc finger nucleases was the first one. Then there were TALENs, and the most recent is the CRISPR-Cas9 technology. So all of them work on principles of a protein complex binding to a particular region in the genome based on the sequences there and creating a double-stranded break. When you do a double-stranded break, what happens? The chromosome is now like floating around like that. The cell has to repair it. So the cell will repair that double-stranded break. And during this process of repairing, it will make some mistakes, right? Those mistakes are what we are banking on. So when you do these double-stranded breaks, you will lead to a large percentage of the repair will work perfectly well, no mutations. But a small percentage will have some mistakes and you will create mutations. Those mutations can then be screened for, and you can then pick up those mutated versions or the alleles where there is a, say, some codon has been skipped, or there's a frame shift, or the other changes have happened. So you basically interfered with the protein's production, right? So you can have a number of different alleles. The protein can be completely dead, or you could have a reduction in efficiency of the protein's function, depending on what kind of mutations you get. So these are genomic changes that you can do to interfere with the protein that is being made from there. And then you go on to look at what are the phenotypes, right? There is one more way in which you can perturb gene function and understand the functions of them. And that is called chemical genetics. So this is similar to genetics, but no genes have been changed. You are only using small molecules. So, and zebrafish is a great system for doing chemical genetics, because the most important reason is that the embryos live in water, right? So introducing chemicals to the fish embryo is very easy. So you can just set up crosses of males and females, collect embryos, and array them into a multi-well plate. So since you have been doing a little bit of experiment during this course, have you seen different stages of embryonic development already? You've seen how small the embryos are, right? So they are small enough that you can even put them in a 96 well plate. So this is the smallest well that you can put in. You can, of course, do that in 12 well or 6 well plates, and you can add compounds. So if I can array embryos into a 96 well plate, what it allows me to do is I can look at the effect of 96 compounds in one experiment, right? It's really high throughput. And you already must have seen that one female can lay hundreds of eggs. So it's a very simple experiment to do chemical treatment of embryos and then look for phenotypes that you're interested in. You might be looking at very gross phenotypes like the shape of the embryo or the size of the eye or something like that. You might also be looking at molecular changes, for which you will have to do other kind of techniques to see whether that you have had an effect or not. I'll show you some examples. Anyway, so in addition to this, you can do live imaging with zebrafish. These are transparent embryos where you can see inside, but the adults are not transparent, right? These have pigment on them, and that means that I can't see what's going on. So once you get used to having this beautiful transparent embryo, scientists got greedy that they wanted the adults also to be transparent, right? They wanted to ask questions in the adults that they could do in the embryos. So they created a triple mutant in 3 different pigmentation genes to get this fish that they called Casper, after the friendly ghost, right? So the Casper fish is transparent except the eye. You can see the eggs in the female, the heart here, the liver here, the backbone, the muscles, everything, all the brain here. So all organs inside are visible in this adult. And this animal was used initially to look at cancer metastasis. So you could actually inject tumor cells into the adult fish, and after a while, you can observe where these cells are migrating to in a live adult fish, right? So these are the great things that you can do using live animals. You must have seen these stages under the microscope now, I assume. So, and this is just a video that Charles Kimmel made in the beginning when people were starting to study fish. And you can see that the first 18 hours is what you see in this video here. There are huge changes that happen in the embryo. From a single cell dividing to hundreds and thousands of cells, them compacting and then moving large distances across the embryo to give rise to a organism that is recognizable with eyes and muscles and a tail and heart, etc. Right? These are the processes of gastrulation, and this makes it a great system to study development because you can watch this live in a normal dissection microscope without any fancy technology involved, right? You can also use a little bit of fancy technology in a fluorescence microscope. If I create a transgenic animal where I fluorescently labeled the heart. So here, the heart is labeled with a red fluorescent protein. Okay. It's a genetically modified organism. Yeah. Yeah. Okay. Right. So in a genetically modified organism, you can actually specifically label the heart muscles. And what it allows you to do is under a fluorescent microscope, you can cut out all the clutter of the rest of the embryo and focus on the beating heart. And this is a live embryo, of course. And you can see the atrium, the ventricle, how it beats in unison. And now you imagine having a mutant where one of this, either the valve is not working or the atrium doesn't beat properly, or there is arrhythmia. You can watch all that under a microscope and phenotype them, right? You can look for genes that are causing these mutations, chemicals that are rescuing these mutations, etc. Okay. Right. And sort of the final thing that I'm going to show in imaging is this amazing feat that scientists did in, I think this paper is from 2018, where they labeled every cell in the embryo. And over time, like I showed you that black and white video, over time, they took fluorescent image, a light sheet image of this embryo, where the cells are dividing, moving, and differentiating into different tissues. So computer, in the sort of the computer, you can actually write the algorithm to label, you can mark one cell and follow it in time. You can see what that cell gives rise to, how many progeny it gives rise to, where do they move to, what do they become later, right? So at cellular resolution, you are watching embryonic development happening. So these are the things that are possible with this animal. Okay. The last one that I want to show, do you recognize the organ? Now that you must have done some work, right? So this is the brain of zebrafish larva. And what you are seeing, these dots of lights that are flashing, is basically calcium firing in the neurons in the brain in a live larva put under the microscope. So this is a fluorescent reporter that has a fluorescent protein that switches on or gives light only when there is calcium bound to it. So what happens when the neurons fire? When is that a surge of calcium is released into the cytoplasm. That calcium then binds to this protein and that cell becomes fluorescent. So you are in real time watching the neurons fire in a live larva. And this larva is immobilized such that the head is fixed in agarose, the tail is free, and you are running a kind of a moving object or something in front of it. So basically, the larva is responding to that movement that you're seeing in front, and the neurons are firing, and you are recording that live, right? So such precision at a cellular level, you will be able to see during this. Okay. So I think I've spoken for half an hour or so. Right. So we have 15 more. I'm going to quickly go over one example of genetic manipulation that we have done and created a model for a human disease. This is what we do. And we have done this for a number of different diseases in the lab. I'm going to talk about CHARGE syndrome here. Okay. So CHARGE syndrome, the name comes from, it's a rare genetic disorder. The name comes from this acronym here for all the different tissues that are affected in this syndrome. It's a birth defect. Babies are born with multiple different problems. And the first year of this baby is a real torture. The number of different surgeries to even just breathe and feed, right? Because there are lots of defects in the craniofacial architecture. So they are not able to feed properly, breathe properly. They have neuronal defects, and multiple different tissues are affected. The gene that is affected in charge is called CHD7. It's a chromatin remodeling protein. What we did is we knocked down CHD7 using antisense oligonucleotide. You will see that written as MO here because these are morpholino modified antisense oligonucleotide. Okay. So this is CHD7 morphant animals. These are control animals. And you see, I don't have to point out also, you can see that there are stark differences in these two embryos, right? And the eyes are smaller, the brain is smaller. This is where the heart is. The heart is not developing properly. And the lower jaw of the animal, which is important for feeding, is not formed at all. When I stain for lower jaw using a marker, I see that there is a severe reduction in the lower jaw cartilages. And what I've put here are images from patients to show you that there are eye defects, there are craniofacial and cartilage defects in the face, and that's what you are seeing in the animal. So the defects that you see are in the same tissues. The fish, of course, never looks like the human, but the organs that are affected are the same, telling you that this gene, it's a human gene, which we found the ortholog in fish and knocked it down, and you see similar phenotypes to what you see in the disease, right? We also looked at the neuronal defects. So this is a disease where the sensory and motor neurons are affected quite severely. So these are the things that happen in the patient. So we look at the cranial ganglia using a marker with RNA in situ hybridization to see where the gene expression is happening in the head of the fish. So this is the head here of the larva, and this is the morpholino oligo injected animal. You can see that there is all the cranial ganglia is severely reduced, right? Here I'm showing you the olfactory neurons, beautifully arranged in a flower shape, all messed up. These are the sensory neurons on the body, which tell the fish about the water current, and all gone, right? These were things that were known from the human studies also. We found some things that were not known. So we looked at the neurons of the fish, and we were just sitting and staring at the fish at different stages in this transgenic animal, which has all the neurons labeled. And we found that the gut neurons of the control, which look like this, is almost completely absent in the morphant animal here. So the gut has neurons on them, and this system, this nervous system, is called enteric nervous system. It's very important for the peristaltic motion of the gut and for the digestion of the food, right? So we saw that there is a complete absence of enteric neurons here, and this was not reported in patients before. But upon searching a lot, we realized that in patients, many patients have constant constipation. And this is a clear sign of enteric neuron defects. After our paper was published, now there are mouse studies also to show that there are nervous system enteric neurons defects here. We also found that myelination of the peripheral neurons is also severely affected in this fish. Okay. So I'm going to skip this part and go to the chemical screen that we did with this fish. So what we did is we saw all these phenotypes. We asked the question, these are a chromatin remodeling enzyme, CHD7. If that is gone, I see all these different defects. So can I rescue these defects using chemicals? And what chemicals should I use? There are thousands of molecules in the world that you can use for a chemical screen. We decided to pick a small number of chemicals that are known to interfere with epigenetic enzymes, right? So we did a small screen of around 30 compounds, and we found, this is how we did the screen, I've already shown you. And we found that there were compounds. So what we did is we took the CHD7 oligo injected animals, and we treated them with compounds in multiple plates, and then we looked at phenotypes. And the phenotypes we were looking at is the jaw that you see here, the eye size, the cranial ganglia, etc. And we found that this is a control. This is the completely knocked down animal and chemically treated fish, which had small rudimentary signs of cartilage coming back in them. And some compounds even had, so this is what the morphine looks like compared to wild type. The ganglia are very reduced and messed up. You see this beautiful recovery in some compound treated animals, which means that even when the gene CHD7 is gone, with a compound, I can recover some of the patterning that you see in the cranial ganglia cells. So this is what this story tells us. Basically, we created a model for charge in zebrafish, and I skipped some part of it, and we used it to used compound to rescue these phenotypes. Right. So this is an example of what you can do, and this is any kind of disease that you want to pick. You can use the same approach, and we do that for various different neurodevelopmental disorders. Now, I want to kind of shift a little bit, and in 5 minutes, kind of introduce you to different models that and studies that can be done using zebrafish to understand neuropsychiatric and neurobehavioral phenotypes, right? So fish is quite a complex organism, and you can do a lot of behavioral studies in fish. It has a social hierarchy in the shoal. There are a lot of interactions. The fish have like hunting behavior and predator avoidance behavior, etc. So it allows you to ask a number of different questions. And there are a lot of different behaviors that has been characterized in zebrafish. And the way you would do it is this, I think you have done hands-on, so I don't need to explain. You can do larval behavior in multiple plates, putting the camera on top to observe how the larva behaves. You can also put the adult fishes into tanks and put a camera on the side to look at the behavior in real time, right? The number of different aspects of behavior that you can address using zebrafish behavioral assays are really wide. So you can look at different anxiety related behaviors, social behaviors, sexual behaviors, sleep has been studied. Even genes that are important for sleep have been identified using screens of sleep behavior. And there is also learning and memory studies that you can do using zebrafish. So just examples. I don't know, which assays have you done in zebrafish in the course here? Have you done any behavioral experiments yet? Social anxiety and social behavior, right? So these are just examples of what you can do. So looking at shoaling, for example, if large number of fish are together in a tank together, they tend to behave as one entity. And this indicates that there is coordination between the organisms, right? If they don't communicate, they would be doing their own stuff. But you see that there is coherence in their movement when multiple of them are there together. If you separate them to zebrafish, sort of using a divider, a glass divider in between. For example, this fish is here and you put a new fish there. This fish has the freedom to go anywhere. To this side, it's a huge tank. But it prefers to kind of come and stay near that glass where it can see the other fish. It's very human nature too, right? To go towards your own species. So this is something that is, so many of these behaviors assays have been developed from mouse behavior studies. They've been adapted into fish. So you can look at social preference. You can look at predator avoidance. So if on the other side, instead of a friendly fish, it is a predator, then this fish would stay as far back as possible, right? You can do learning experiments where you can put them in mazes and teach them that the food is given on one. Does the fish go towards that food over a number of different trainings? And we do that too. You can look at anxiety, which you have already done, you said. So for example, this is an sort of a real life example from our lab where we have stressed these fish severely and put them into, so the fish is put into a new tank. So what happens when you put a fish into a novel tank, like many other organisms you can imagine, it would first go to the safe place, which is the bottom of the tank, stay there for some time. When it feels a little more comfortable that there is no threat, slowly it will venture upwards to explore the top of the tank. So this is what you see in this fish. Okay. So this fish has finished its sort of time here, and now it is gone up to explore. This fish has been through a lot of stress. So this fish does not dare come up because it is in the high stress and highly sort of anxious state, and it sticks to the bottom of the tank. And you can measure this using a camera and do a tracking. So you will see this sort of sticking to the bottom in the beginning and then exploring on the top. If it is very highly stressed, it will stick to the bottom only. And if it is, you give something to it that makes it fearless, it'll start doing this. Instead of staying at the bottom, it will go up, risking everything, right? So these are the kind of things that you can do to look at neurobehavior in fish. And you can imagine creating models for various different diseases, anxiety, schizophrenia, even neurodegenerative models have been created in zebrafish in different kinds of contexts. So you can do it genetically as well as environmentally. You can mutate genes that you suspect are important for social behavior, for example, and see what happens to the fish. Or you can give it an environment that causes stress, or a drug that causes stress or overactivity or sedation, etc., right? And study the behavior in fish. And that is a beautiful correlate to the human behavior that you see. And this is a simplified version, so easier to study. I will end with a couple of different resources that for those who became fascinated with zebrafish from this course that you're taking, you can go back and look at this page called ZFin. So a lot of resources are there, labs are there, genes, their expression patterns, the genome sequence of these genes and what they correlate with, etc. There is also this site that is just a, even if you're not interested in the brain, it's just so beautiful, this the images on this site. This is the zebrafish brain atlas, and this is the front page. And each page, if you click different regions of the brain, they have imaged different transgenic animals. So here you are seeing a triple, quadruple transgenic animal. One that is purple labeling all the neurons here. The green ones are labeling the axon tracts. Then you see this midbrain, hindbrain boundary, I think, which is labeled with a different gene here. So you see this multiple colored images, which makes it very easy for you to identify different regions of the brain, also different kinds of neurons in the brain. So these lines can be used for asking molecular questions in detail of which neurons are important for which behavior, and doing a lot of beautiful experiments in the neurodevelopment area. So I'll finish with, this is my lab over time, and I can take questions. Yeah.
So you showed it, right, where the vision for the drugs where many questions can be screened for what particular type of drug. I was just wondering, like, you want to study the effect of drug on different classes of neurons at the very same time. Can it be done?
So having these kinds of transgenic animals where different neuron groups are labeled, you can use them, put drugs on the fish and look at effect of it on the neurons. You can also then, I guess, I did not talk about optogenetics. So something that I missed out. Has anybody heard of optogenetics? So what you do in optogenetics is basically you express a channel rhodopsin in particular groups of neurons. And these channels are responsive to light. So you can shine light on them, the channel will open, or you can shine light on them and they'll close. So if you want to ask this question, for example, are these particular group of neurons important for predator avoidance in fish? So you express the channel rhodopsin in that group of neurons, and then you shut it down or activate it and see how the fish behaves in response to predator. So this way you can actually connect neuron to behavior. So at molecular level. So you can do this. These are done in first experiments were done in mouse, but in mouse you can imagine you have to open the skull, insert a laser, fix it on the mouse, and then do the light shining and all. Here you just have to put it under the microscope and do the laser shining. So it can be done very easily because also it's transparent. So none of the hassles of this. So you can actually do this kind of experiments. Yeah. So there are a set of behavioral assays you can do on larvae. There are a set that you can do on adult. You can yourself imagine that there will be things that the larva will not be able to do that adult can do because it's a more complex organism, right? Also, there are things that you cannot measure in the adult because it's a large animal that's moving around. While the larva can be fixed on the under the microscope and you can make it do things and observe it in live animal. So yeah, there you can do behavioral assays in both, and there are different sets of questions you can ask with. Yeah.
Uh, Good afternoon, ma'am. My question is related to CHD7 and Charles syndrome. So, in when you injected these different molecules, not molecules, chemicals. Some chemicals did show some improvement. So, what exactly like in Charles syndrome there is this CHD7 gene is becomes absent or becomes
It is
inactive.
mutated. So it is a dominant disease, which means only one copy is mutated. The other copy is wild type. Even then, the patient has such strong phenotypes because the levels of the enzyme is very essential. If the level of the enzyme goes down, you get a phenotype. This is called haploinsufficiency, right? So yeah.
So, the chemicals which are showing good results, what do they do? Like, do they have functions similar to the protein which the CHD7 codes?
So the CHD7 is a large enzyme that opens chromatin. Okay, so it is a helicase that opens up chromatin. The compounds that we used, some of them are, for example, HDAC inhibitors. Histone deacetylases, when you inhibit them, the chromatin becomes looser in a very crude sense. So these compounds affect the epigenetic landscape, the chromatin shape in the cell. The exact molecular function is not the same as CHG7. What the molecules are doing are compensating for the action of CHG7 by opening or closing or acetylating or deacetylating the chromatin.
Thank you, ma'am, for providing us with valuable insight. Now I request our moderator, Dr. Sunil Kumar, to facilitate our speaker with the memento. Thank you.
Yeah, my email address is there, and you can find me on the website. Happy to answer questions.
Okay, thank you for coming and
I just want to add that superfish is something that does not require big technology. So even in schools and colleges, we can do different extent because a lot of it is observation. Right? So
quite
sorry
often
for
our area
rushing off.
is learning and learning. And one has to come out of your comfort zone. We learn many things, but then we go out. Okay, so you have another one? So we have to start with the in fact, okay, so now there is 20 minutes the other biolab people that has got to break again, it's here in my office, I guess. What anything that you have tomorrow there will be a feedback or there will be circulated. Yes, so you have to fill it up and deposit it tomorrow. I mean, you have to do it by like tomorrow morning. Of course, it is full form of something you're right there. So why I am doing it by today itself, I understand. Because tomorrow there is more time there. Because again, 9:30 onwards, one for my sister have your program. From my very science department, then we have Arnav from NII, and then we are the director, which is talking to that. Now, check ready with this talk, then you will.
So please have a seat. I'll give a brief introduction.
Yeah.
Now, moving further, we have an industry talk given by Dr. Prashant Sharma, application scientist, BioRed, who are also the sponsor for the workshop. He has been associated with BioRed for more than a decade and has a vast experience for various genomics, proteomics, and molecular biology techniques. I kindly invite him to step forward and offer us his insightful words.
Thank you very much. So, first of all, sorry for being late. Actually, there was a lot of traffic in the service lane, so it took time to enter actually. So, I'm not aware that which techniques actually you have seen in last 3-4 days actually in workshop. So, we are covering today electrophoresis and Western blotting. I hope you have not covered it. Ma'am? So, this technique was not covered. Huh? This technique was not covered in already covered. Okay. So, if it has been already covered, then can anyone tell me about Western blotting little bit? It is not covered. Okay. So this mic is working? That is not working. Okay. So first of all, let me introduce myself. So like ma'am has already introduced, but just in a brief. So I'm working with BioRed since last 10 years and working as application scientist and taking care of all the training part of BioRed instruments. So whoever is buying BioRed instruments, they are getting trained by our team. Yeah.
Just introduce the thing. No, they have not done BioRed. They have been working with Western blotting.
Yes, I
Because
understand.
some protein they have been separated. The idea is you are the expert, as you are rightly saying.
Yeah.
You give this thing, but in the basic in 15-20 minutes, all right?
Yeah,
And
sir.
if
Yeah.
there is anything, you will show.
We will discuss
Because
it.
we are waiting for you to come and they have separated the protein and now they will run the check.
Okay,
So we
good.
have.
Great,
planned
yeah.
that you are
Great.
coming with the experts view, therefore everybody will say that, or see that, all right?
Okay, sir. So we'll try to finish in within 20 minutes.
There.
Yeah.
Expert than many other people.
Oh, okay, sir. So that's why I have just kept the topic key aspects of optimization, because like Western blotting is not a big thing actually doing, but there are multiple points actually which you should take care actually when you are doing experiments. So those part we will discuss. So first of all, what is the use of Western blotting? So like these are the two main objectives doing the Western blotting. First of all, to determine the presence or absence, like whether it is positive or negative, kind of that, like band is visible or not. Second is to quantify. Okay. So for quantification of any protein, you can just do Western blotting. And this is for a particular protein, not a whole solution actually. Okay. So this is the workflow. If you see proteins this is not workflow. This is about how modification happens actually inside a cell. Okay. So like in protein synthesis, then protein post-translation modifications, then subcellular localizations. So like in every stage, the protein, there are some changes are being done actually inside the protein to work in different pathways and different organelles actually. So actually, when you are doing any experiment, you should be able to isolate that protein which you want to identify or which you want to quantify actually. So sometimes this is a mitochondrial protein, sometimes it is a nuclear protein, or sometimes cellular protein, like cell wall protein. So that protein should be soluble in your solution, first of all. So before doing any isolation, you should be aware that which protein you are isolating and that is soluble or that is coming in the solution or not. So further we will see other things. Okay, so before you begin, these are the few points actually which you should consider. First thing, process. So what controls should be included? Okay, so like in any experiment, you run positive control, negative controls. Okay, so here also there are some controls which you should consider. Okay, so like there should be a control where you are not giving any treatment versus you are comparing a control or like sample. Then loading controls are there. Loading controls means when you are loading samples actually. So when you are loading in multiple wells, there should be what to say, measurement that you are not loading different volumes from different wells. Suppose you are running 10 wells and in all 10 wells you are loading different volume, then finally you will see expression variation actually in the loading control should be there. And then positive controls, antibody validation you should do before starting your experiment, that which antibodies you have to use. And then process validation, like whatever method you are going to use, that will work for your experiment or not. Then measure experimental error with technical replicates. So like you can run same gel or same sample in multiple gels, and then you can run multiple blots just to verify things. Okay, create a statistical significance means like you should run samples at least duplicates and triplicates, and you should include more number of samples actually just to make your study valid actually. Suppose you are running any screening thing and you are running only 3-4 samples and it is not sufficient. You should take bigger group actually, more number of samples. So ultimately this is the workflow for any Western blotting experiments. And in Western blotting you can use chemiluminescence, fluorescence, and colorimetry also. So there are different ways, and different labs are doing according to the instruments which are available in their lab. So first step is sample preparation. Okay. So sample preparations means whatever proteins you want to run actually, that should be in the solution form. Okay. So like suppose you are using E. coli grown in media, or suppose you are taking some tissue or something which you are lysing and then you are isolating protein. Okay. Or sometimes you are isolating proteins from plant tissue or any leaf or something. So that protein should be in a soluble form. Then you have to run that protein on a gel. So for running a gel, actually we do electrophoresis. And in electrophoresis, which kind of gel we run actually for protein electrophoresis? Polyacrylamide gel. Okay, so that we call SGS phase. Okay, SGS phase technique basically, and where we run polyacrylamide gel. Okay. In polyacrylamide gel also there are different types actually. Sometimes you are running native phase, sometimes you are running SGS phase. So like one is denaturing phase, the other one is your non-denaturing phase. Okay. So according to protein and which type of function you want to establish, you have to select the gel. Okay. Then after running the gel, you have to do transfer. So transfer is what? Whatever proteins you have separated in the gel, those proteins you have to transfer on a membrane. So why we are doing transfer actually? If proteins are already separated in a gel, can we not detect those proteins in the gel with the antibodies? So it is very difficult actually to do all that process in the gel. And gel is fragile, that can break during processes also. Okay. So it's better to stack that protein on a membrane. So here we use nitrocellulose membrane or PVDF membrane. Okay. So the binding capacity of the protein is different according to the membrane type, but we usually use PVDF or nitrocellulose membrane. Okay. So that membrane we have to use, then protein will be transferred from gel to membrane. So this process we will see here in the lab today. Okay, like how to run a gel, how to transfer proteins from gel to membrane, how to visualize those proteins actually. And then finally, when the protein are transferred to the blot, then you have to start the antibody treatment. So antibody treatment means like you have to first incubate with primary antibody, then secondary antibody, and then finally detection. So that process is actually antibody part. So how to select the antibody, first of all, so that was part of process validation and antibody validation. So first of all, you should be aware that which antibody you are going to use, which secondary antibody you will use, which method of detection you will use. Accordingly, you will start buying consumables and starting your experiment. Okay. So this primary, secondary antibody, and then which conjugate method you are using, like you are using any colorimetric method, NBT-BCIP method, or you are using chemiluminescence like HRP method, or you are using fluorescence. Okay, so there are multiple methods of detection of the protein, and sensitivity of each method is also different, but it depends that which technique is available in your lab. Okay, many times, many customers are doing fluorescence, but for fluorescence, you need at least a chemidoc instrument which is capable of doing that. So after doing Western blotting, finally you have to capture the blot. In blot, you have to capture the signal, and from that signal, you have to quantify expression level of the different proteins in different samples. Okay, so this whole process we can see today. So how to like first of all prepare your sample. So for preparing sample, you have to do lysis. So lysis means you have to grind your sample like in a fine paste or like very thin type of solution. Okay, and then you have to do solubilization of the protein. So for solubilization of proteins, like anyone has done any protein isolation? Sir was telling that you have already isolated proteins. So like how was that sample prepared? With lysis buffer. So, okay, so which type of sample you have used? Brain sample. Some neural tissue, basically. Okay. So, first of all, you have just done some grinding or something just to break the, for homogenization purpose. And then you have solubilized the protein in the solubilization buffer or lysis buffer. Okay. So, that solubilization part is basically solubilizing all the proteins. Like sometimes cellular proteins are not soluble, like cell wall proteins are not soluble easily. Okay, so for solubilization of that thing, you need some special conjugates or special chemicals like Tritonex 100 or some other conjugates. Which chemical you have used? SDS buffer and EDTA. Yeah, those are basic things, but some special conjugates are also required actually to solubilize those things. Okay. And after solubilization of the protein, you have to remove contaminants. So for removing contaminants, how to remove actually contaminants from solution? Centrifugation. So with centrifugation, you can use like, you can remove all the debris part. So whatever non-soluble material that you can remove just by centrifugation method. Okay. And after centrifugation, whatever supernatant is there that you are collecting, and that supernatant part actually, you are just checking the protein concentration. So for checking protein concentration, you are using UV visible spectrophotometer. So this picture is basically a smart spec instrument from Bayer, but that is also UV visible spectrophotometer. Okay, and finally you are running that sample on a phase gel. Okay, in between there are multiple steps also involved. Suppose you want to enrich your sample. Suppose you want to remove all the other contaminants which are soluble also and which can interfere protein electrophoresis during like gel run. So like suppose DNA will also be there. So suppose you want to remove DNA, then in that case we can do protein precipitation method. So in that case only protein will be precipitated. Rest all contents will remain in the solution, and we can just discard the supernatant, collect only the pellet, and re-resolve that pellet into a buffer actually. So suppose like loading buffer or any other solubilization buffer in which only protein is there. So this is the method of enrichment actually. Okay. So there are multiple steps involved. So how do we run actually? So we cast a gel, and on a gel actually you are running your sample. So this is just like some bookish thing like how to prepare a gel and how to run. So that process we will see. So I'm not taking much part on that. Okay. So gel running, I think you all must have done. SDA space part, you already know, I think. Okay. So SDA space, I'm not explaining that much. We will see that what are the things actually we should care about when you are running. So after running a gel, now how to transfer it on a membrane. So for transfer purpose, you need a transfer apparatus. So there are like total wet transfer method, semi-dry method, or total dry method also, like where we are using only 5 to 10 ml buffer only. So it depends on like how much molecular weight protein you want to transfer. So the range actually. So sometimes when you are just transferring low molecular weight protein, then you can use semi-dry or dry method. But when you are using like a high molecular weight protein, then you need a technique through which you ensure that all the proteins are eluting from gel and just reaching up to the membrane, okay, or binding to the membrane. So like this is one method, one of them. This is also semi-dry method, but it is very fast actually. So very fast means like if you are using only bioride conjugates through whole process, then you can transfer proteins within 3 minutes also. So like there is some high concentration buffer we are using through which all the proteins are eluting very quickly and just transferring to the membrane. So this instrument I just brought with me. So we will see this part today. So after transfer actually, how do you ensure that transfer has been done? By Ponce staining. Okay, so by Ponce staining, usually we like stain the membrane and then just see that whether protein transfer has been done or not. Okay, so many times protein like people like different you just take pictures of this Ponce stain also, and they use this picture for normalization purpose. So do you know normalization? Yes, sir. So like, yeah. So when you are running your samples, okay, in different bells, and you have loaded equal amount of protein, okay, even then we can't control many things actually. Even then variations will be there, like during electrophoresis itself, lot of variation will be there. So like in one well, you have loaded suppose 20 microliter, in other well by mistake you have loaded 21 microliter or 19 microliter. Just because of that itself, there will be variation. To control those things, actually, we always detect housekeeping protein. Okay, housekeeping proteins or reference proteins. So what are housekeeping proteins basically? These are the proteins whose expression is constant throughout the different conditions. Means suppose you are exposing some culture at room temperature at 37, at 50 degrees Celsius or at 70 degrees Celsius. Okay. So during all temperatures, there will be differential protein expression for few proteins. Okay. Just to survive that culture. Okay. So, but there will be some proteins which are constant even in all four conditions. So we have to take those proteins which are constant throughout all the conditions, and we use those proteins actually for normalizing your samples. Okay. So suppose there is a loading variation that can be omitted just by using housekeeping proteins. Okay. So many times like people have used Ponce stain picture also just for normalization purpose. So like here we take total lane intensity, and with that we compare actually the your target protein expression. So if suppose in a lane, whatever protein sample you are loading, suppose you have loaded 20 nanogram or 20 microgram in each lane, then total lane intensity should be same because you are loading same amount. Protein concentration can vary, but total intensity will be same. So, okay, so just because of that intensity should be same, so we can derive a normalization factor, and with that we can multiply the target protein expression. So this is also one method actually to detect, but this is reversible method. Like after washing with water, you can remove the sponge stain and you can use this blot for further purpose. Okay. So do we perform blocking before Ponce stain or after Ponce stain? After Ponce stain. Okay. Because if you block it initially, then whole blot will look pink actually. Okay. So there is another method which we use actually in Biorad. So this is stain-free technology, and we are promoting it a lot because this can reduce lot of steps in Western blotting, and this can give you very reproducible results also. And this is not a new thing. This is already JBC recommended thing. Like JBC has already recommended to users that they can use this technique. So what we do, we are using one special compound which binds with most of the protein. Okay. So when you are running a gel, the same compound during running of the gel is binding with proteins uniformly. Okay. So when like during run itself, if something is already like bound to protein, you can visualize that just after gel run. There is no requirement of any staining. So like what usually we perform after gel run, either you have to stain it with Kumasi blue, then only you can visualize, or you need something through which you can visualize without staining. So that is that particular thing. So when you are using stain free technology, there is no staining required. So like just after running the gel, you can just put it on the UV, and on UV itself, you can see that how the protein separation was done actually. So when we run agro gel, in that also we put it on UV only. So in case of protein also, you can see proteins separation just by UV. Okay. So by running this technique, actually, by using stain-free technology, you can use same gel for blotting also. And after blotting also, you can see that how much protein has transferred from the gel to membrane and how much protein left in the gel. Okay. So this is a way of verifying multiple things and omitting multiple extra steps. Suppose if you don't use stain-free, then you are using Ponzi staining. Okay. But here with the stain-free, you can directly visualize. Suppose you want to use this stain-free technology for normalization purpose, then you have to just take picture of the gel, and that picture you can use for normalization. There is no need for detecting additional housekeeping protein. Okay. Because you have total lane intensity for each well or each lane, so that intensity can be used for normalization purpose. So that we will explain in further slides in more detail. So how it works actually. So you can see on the right side, the picture is for a blot. On the left side, this is total protein of same gel actually. Okay. So you can see that in different lanes, suppose in this lane, protein concentration is more. So few ones are looking very like dark and few are very thin actually. But when we do experiment, we usually load same amount. But even then, if this kind of variation is there, it means something has happened during running the gel, or by mistake you have loaded, or maybe the quantification which you have done for protein sample, that was not that much accurate. Okay, because you can just this quantification is just for getting the range actually. That is not very much accurate. Suppose you are like whatever method you are using, if it is telling you 20 microgram per microliter, that is not accurate 20 microgram. It can little bit vary. Okay. So this concentration of the solution, or just because of loading, you will see this kind of pattern. Now, suppose if you have already loaded some extra amount in this particular lane, then definitely you will see thicker bands in the blot for that particular lane. Okay. So how to normalize? We have to just take lane intensity for each lane, and for first lane, suppose we are taking reference. So we have to just divide first lane intensity with other lane intensity just to drive a normalization factor for each lane. And that normalization factor will be used to multiply in the total signal of particular band in particular lane. That way you can normalize. So there is no need of using any housekeeping protein. Okay. And this part is like pre-enabled in Bio-Rad software. So suppose if you are using Bio-Rad workflow, then you can easily use this technique. So now after like doing blocking and Ponzi stain, or suppose stain-free imaging, you have to just block the blot. So in blocking, what we do? Skim milk or BSA. So like you have to block the area of the membrane which is unexposed, or like which is clean. Clean means there is no protein. Okay, because if you are not blocking that membrane area, then what will happen? Non-specific binding. Okay, so like the active membrane is there, but if that is free, that area is free, then whatever antibodies you are using, those antibodies can bind to that area. Okay, so those antibodies binding to that area, then they will give non-specific signal. So good blocking is very much necessary. Okay, so for blocking purpose, either you store your membrane in skim milk solution or BSA solution just to block full membrane. And there are different experiences with different solutions. Sometimes BSA works good for your experiment. Sometimes Skim milk works good for your experiments, and it takes a lot of time also in blocking. So for that purpose also, we have something like every blocking buffer through which you can just block within 5 minutes. So just 5 minutes, you incubate your membrane, full blocking will be done. Okay, so that is also one solution from Biret. So after blocking, you have to incubate membrane in primary antibody solution, and this depends on the antibody concentration, and your target is very low expressed. So the timing or the incubation will vary according to that also. Sometimes it will do only 1 hour, 2 hour incubation. Sometimes they do overnight incubation also in primary. So it depends due to experiment types. And after that, you have to do washing and then secondary incubation, like secondary antibody incubation. So here we are using secondary antibody labeled with some enzyme actually, or some fluorophores. Okay. And when we are providing that substrate is being converted to some other molecule, and it is giving some fluorescence or some chemiluminescence or some color actually. Just because of that, it is detectable in different instruments or some detectors. So for taking a picture of any gel or blood, which instruments we use? Gel docs or chemidocs. Okay. So suppose if you want to do chemiluminescence or fluorescence, then we use chemidoc. For normal gel applications, we use gel doc. Okay. So these are different steps. Then secondary incubation, then washing, and then finally detection. Okay. So there are different molecules which we can use, or different methods. Like this is colorimetric method, chemiluminescence method, and these are the basic principles which whenever we will have time during workshop, I will show this presentation there. You can just discuss about it. Okay, there also. So this is fluorescence, how it works. So like colorimetric, chemiluminescence, fluorescence. There are three methods. And uh, according to the method, the sensitivity is also different for all three methods. Okay. Now, so this is chemidoc instrument. The example of chemidoc, basically, the dynamic range of this instrument is like very good, and yeah, yes, sir. So now these are the basic advantages of fluorescence imaging. So like wider dynamic range, more accurate quantification, and ability to multiplex. So suppose you are running a blot. In a blot, you can detect only one protein. Suppose if you are doing chemiluminescence, so when you are treating that blot, on whole blot, you can just detect one protein, okay? Because you can use one antibody for primary and secondary, okay? Suppose you want to detect multiple proteins. So in that case, you have to use a multiplexing technique, which is fluorescent Western blotting. So in fluorescent Western blotting, what we are doing, we are using primary antibodies labeled with different fluorophores, or secondary antibodies labeled with different fluorophores for different targets. This way, actually, you can detect in a single blot, you can detect 2 or 3 proteins. So one protein, suppose you want to use for normalization purpose, housekeeping protein, and remaining 2 will be your target protein. Or suppose if you are using stain-free technique, then for normalization, you will use stain-free, and remaining 3 will be your target protein. So like that arranging sample itself is difficult, then making sample again takes time, and sometimes the sample is also very limited, that you cannot arrange that sample again and again. So it's better that you adopt a technique in which you can do more, you can create more and more results just with limited amount of sample. So just with fluorescent Western blotting, you can detect more and more proteins actually. So, so on the right side, actually, you can see there are 3 color shown, like yellow, green, and red actually. So those are basically for 3 different proteins. So like all proteins are in a same blot at different molecular weight range, but all are being detected in a single blot itself. So, going sort of time. So we can finish it here actually, and then uh, when we are doing practical, actually there we can discuss it more. Thank you very much.
Thank you, sir, for the insight. I would like to request our moderator, Dr. Sunil Kumar.
Just one picture, unlike many of the theory pictures, but there is This is just to give some example, and we are grateful of their thankful that they have been here for a while. And they, since they are expert, they know much more than many of others. But of course, many researcher or postdoc or working like many of the same. But without wasting time, we should go. Yeah, so there are two of them. Like, okay. So thank you for the yeah. Okay, so now we go to the lab. The lab give us 10 minutes so that question शायद तो भी है या आप कभी होता है हाँ ठीक है तो then they can so you want to be right away because you have been coming traveling or you follow जब आए हाँ जब आए you have water here so when you say things, the one that you said, how does it work? You add something in the buffer. You don't get it. We add it in the buffer. So you have to buy the buffer also from you. I requested we have yesterday also this was asked. We can touch or rather 2020 I'm not sure. Committee this we acquire a journal, Western process, not the journal of our you might have readings, but it's what journal tomorrow we are getting you whosoever has supplied, you see our situation. यहाँ the system and whatever they do, that you go there and somebody tries to give you to discount go this that. You make it one manner that you give us something frozen we are there. आप अपनी तरफ से discount होता है, वो हम अब करते हैं. You see by sending somehow आप करते हैं, लेकिन तो हम लोग करते हैं. Okay, as a you also passed out from there. Add something. Each one project become input by the concept. This is my request because when I am in the lab, we diet, we talk, negotiate, hear what they do. They think they know everything better than us. So, if I just make something, you give me some discount after that, the purchase department will say the customer. Therefore, what last one year after they don't keep supposing keep 10%, go 5%, they get 5%. Paisa way the something so that I can but I tell you who has given that because somebody must be responsible writing a sale in the part. So but tomorrow the system will approve and then pass it that. But I know you are technically they are consistently but they are your friends, right? So after my laptop on yeah. So let us go there in the So you are Mr. Prashant Kumar, you are Dr. I am Nitesh. Nitesh. So did you ever come to change or that? Who used to come here? Because we have. I haven't been here now. Quite difficult here. Earlier, the person used to come, some Afghani or something, who always used to come. Who was manager or something. Maybe 15 years back, or 20 years back, I used to do. I think I remember I back in 2007. Around that time, or even before that. So those people used to come. After that, the students used to handle for people like this. I never knew exactly who is coming, who is where is it. Nobody someone saying chicken. This is government. So today is morning, they have prepared. See, the point is, there are 20, we wanted 50, but then their applicant is 24, but they are coming out to 21. Right? Everything we cannot, these are behavioral behavior, it's not only protein estimation. It's one part. Even if we are taking out what type of behavior that is also for the study, they will do along with you people, almost everybody will be taking this. The other things, there are only 7, 8 type of things going on last week. Some behavior, some electrophysiology, right? Brainwave analysis, and the computational biology, so they get there up, right? And that way, my lab is also started on it. Particularly, this is neuro. Yes, this is a neuroscience institution. So everything is on neuro. Be from molecule to biochemistry to behavior to electrical signaling to this is the first thing that is not the problem. इसका क्या एक्स्ट्रा ये रिकॉर्ड हो रहा है क्या? खत्म हो गया
तो माइक ही नहीं जाएगी क्या?
हाँ
अच्छा।
हेलो, ये इसका प्रोग्राम खत्म हो गया सुरेश का, सुरेश वाला, जेटू वाला, जीवन वाला खत्म हो गया यार, कब का खत्म हो गया? अच्छा, पूछता हूँ मैं मुझसे।